We will be describing the stages of mitosis. An interphase is where the cell spends most of its life. There are three main phases that can be explained, which are G1, S phase, and G2. After the cells have finished dividing their chromosomes and cytokinesis has divided the cell membrane, the two cells enter the first stage of interphase, which is G1. During this stage, the cells perform its normal function and it grows. In addition, there are cellular organelles in cytoplasm, including important proteins and other biomolecules that are duplicated. In the S phase, this is the point at which the DNA is replicated. After the DNA has been replicated during the synthesis stage, the cell enters a second gap stage known as gap 2. During G2, the cell adds volume to the cytoplasm and replicates many important organelles. G2 is spent double checking that no errors have been made during DNA replication.
Prophase is the first phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During prophase, the complex of DNA and proteins contained in the nucleus, known as chromatin, condenses. The chromatin coils and becomes increasingly compact, resulting in the formation of visible chromosomes made of a single piece of high organized DNA. The sister chromatids, replicated chromosomes that have an X shape, are pairs of identical copies of DNA joined at a point called the centromere. Then, a structure called the mitotic spindle begins to form as a result. The mitotic spindle is made of long proteins called microtubules that begin forming at opposite ends of the cell. The spindle will be responsible for separating the sister chromatids into two cells. Prophase is when the nuclear membrane breaks down into numerous small vesicles, allowing the spindle microtubules to have direct access to the genetic material of the cell. As prometaphase ends and metaphase begins, the chromosomes align along the cell equator or metaphase plate. Every chromosome has at least two microtubules extending from its kinetochore, which are attached to the spindle poles at the opposite ends of the cell.
Anaphase is the fourth phase of mitosis, the stage after metaphase, where the replicated chromosomes are split and the sister chromatids are moved to opposite ends of the cell. Anaphase begins when the cell passes the spindle formation checkpoint, which allows chromosomes to separate. The kinetochore microtubules are responsible for dragging the chromosomes, while the spindle microtubules push the two centrioles apart. Once the chromosomes are drawn to each side of the cell, the non-kinetochore spindle fibers push against each other, stretching the cell into an oval shape. Anaphase accounts for only 1% of cell division.
Telophase is the fifth and final stage of mitosis before cytokinesis. At this point of mitosis, the cell is nearly done dividing. During telophase, the sister chromatids reach opposite poles and reform around the group of chromosomes at each end. The mitotic spindle is an assemblage of microtubules and proteins and is broken down into its building blocks during telophase. Any remaining spindle microtubules are depolymerized. Two nuclei form, one for each set of chromosomes, and the nuclei begin to reappear. The chromosomes become less condensed and return to their original stringy form. At the end of telophase, the division of one nucleus into two identical nuclei is complete.
Cytokinesis is a stage in cell division where the cytoplasm of a single-cell eukaryotic cell divides into two daughter cells. During cytokinesis, the spindle apparatus partitions and transports duplicated chromatids into the cytoplasm of the separating daughter cells. It ensures that everything is maintained from generation to generation and that the daughter cells are effective copies of the parent cell. After this stage, each daughter cell begins the cycle over again and enters the interphase of the cell cycle.